Okay, so as I mentioned at the end of the last video lecture, we keep talking about the myocardium and how important its function is. You know, we just took a look at the blood supply to the myocardium. Now we're going to zoom into the myocardium. You know, myo means muscle, so we're going to be taking a look at this cardiac muscle tissue that composes the vast majority of the walls of the heart itself. All right, so if you take a look at the microanatomy or the microscopic anatomy of cardiac muscle tissue, now if you looked at it under a microscope, it would actually look like this, and your cardiac muscle cells or muscle fibers kind of look like skeletal muscle fibers. They're long cylinder-shaped cells. That's why a lot of times they're referred to as fibers. And what's different, though, between those and skeletal muscle fibers is that they have branches. So like here's a muscle fiber up here, here's another one over here, and then you'll have these little branching areas like this that connect that cell to another one there and another one over here. So that branching takes place. The cells are not nearly as long as your skeletal muscle fibers. They tend to be shorter. They're connected to each other through features that are called intercalated discs. Intercalated, where it kind of has like a zigzag border between the two. That's what that's referring to. And you may remember from Biology 201, your skeletal muscle fibers can have lots and lots of nuclei inside just one cell, and your cardiac muscle fibers, they're not as long as a skeletal muscle fiber. So they typically have just one, maybe two nuclei, and those are like, there's one right there in that picture, and they tend to be centrally located. Okay, there is your cardiac muscle fibers also do have some little thin strips of connective tissue, also known as an endomysium. Maybe you remember that term from your Biology 201 as well. Endo meaning within. The endomysium are these very thin, delicate connective tissue coverings around your skeletal muscle fibers. And you have that as well in your cardiac muscle. And that connective tissue connects to something we call the cardiac skeleton. Those are tougher connective tissues within the heart that help form borders between the atria and the ventricles, and also the big blood vessels that attach into the base of the heart, where all those big blood vessels are connected. You know, those are composed of connective tissues as well. The connective tissues provide pathways or little thin fillers for the capillaries, the microscopic blood vessels that are going to be bringing the blood supply to these cardiac muscle cells. So actually, let me erase that for a second. On this diagram, do you see how you kind of have the lighter pink areas in here and over here and over here? The darker pink striped looking cells, those are the cardiac muscle fibers. They look striped, and that's referred to as striated. You may remember that from Biology 201 as well. And those lighter pink areas, what you're seeing there in that diagram, this picture that was taken with a microscope, those are actually the connective tissue fillers in between those cardiac muscle cells. All right, and then your cardiac muscle cells are joined to each other through features that are called intercalated discs, where you kind of have this wavy junction. So you got one cell over here and another cell over here. And this is different from what you see in skeletal muscle fibers. They don't have these intercalated discs. They're kind of shaped like corrugated cardboard. You know how corrugated cardboard has kind of a wavy surface like that, and it actually makes it stronger when it's corrugated, because if you have two pieces that are linked together in these waves, it helps hold them together. That's the same thing that's going on with your cardiac muscle fibers, because where one cell is connected to another through these waves, these intercalated discs, that helps hold those cells together, which is kind of important if you think about it with your cardiac muscle fibers. Your myocardium has to beat all throughout your lifetime, And you want those cardiac muscle fibers to be held tightly together. And so these corrugations or intercalated discs help that happen. Within there are a couple of terms that you will hear about or read about within the intercalated discs, these little wavy connections between cells. You have some features that are called desmosomes, and those are basically proteins that are located at those separations between the cells that they function like cell snaps. They help hold those cells together. If you look closely at these diagrams from your textbook, it's showing you that kind of looks like protein snaps helping to hold the cells together. And then the other feature that's really important are the gap junctions, and those are pores that exist between individual cardiac muscle cells. And what those do, they provide pores through which ions can pass from cell to cell. Why is that important? Well, ions, charged, little atoms that have positive or negative charges, are super critical for the electrical activity of your heart for generating those action potentials. Remember those from studying skeletal muscles in Biology 201? Well, we have those again with cardiac muscle fibers. And so you've got to be those in the heart. Those gap junctions allow ions to pass freely from one cardiac muscle fiber to another, and that allows action potentials, these electrical changes around the cells of the heart, to flow very quickly from one cell to another, to another, to another. When you study the skeletal muscle system, you learn that your skeletal muscle fibers are all well insulated from each other, and skeletal muscle fibers, if you have an action potential on one, it doesn't spread to another skeletal muscle fiber. They each have to be stimulated individually in order for a skeletal muscle fiber to contract. Not true with your cardiac muscle fibers. Your heart beats as a unit, and there's a term for that. It's functional syncytium or syncytium. You'll hear people pronounce it that way as well. What that means, that phrase means that the heart functions as a unit. So your cardiac muscle cells beat together in unison. In order for that to happen, you have to be able to allow an action potential, those electrical changes, to spread very quickly from one cell to another, to another, to another. And that's able to happen because you have these gap junctions, or little pores, that allow ions to flow from one cell to another. Okay, and here's a pretty nice diagram from your textbook, which is blowing up a couple of cardiac muscle fibers for us. And like over here, here's one cardiac muscle cell or muscle fiber, and there's an intercalated disc between this cell and the next one over here. And here, this has been cut open so we can see the border between one cell and another. We've got some gap junctions. You would have some desmosomes in there helping to hold that cardiac muscle fiber to the one next to it. And then here's one of those branches that is extending to yet another cardiac muscle fiber over here. And they're cutting this open. They're showing you in your cardiac muscle fibers, you have myofibrils, just like you learned about in Biology 201 for your skeletal muscle fibers. Those myofibrils are full of actin and myosin protein filaments, and you have the sliding filaments. The actins and the myosins form connections with each other, and they slide across each other, and that allows your cardiac muscle fibers to contract, just like what you learned about in Biology 201 with your skeletal muscle fibers. Also notice mitochondria. You've got lots of mitochondria inside your cardiac muscle fibers. And what do mitochondria specialize in? They do a lot of cellular respiration, which makes your ATP for energy. So we need a lot of those in our cardiac muscle fibers as well. All right, we are not going to go in. I know you guys are going to be crushed, but we're not going to go into the gory details of how action potentials are generated on your cardiac muscle fibers, not nearly in the kind of detail as what you had for Biology 201 when you learned about your skeletal muscles. But we are going to hit some of the highlights here, and we're really going to focus in on the differences between what goes on with your cardiac muscle fibers versus your skeletal muscle fibers. All right, one key difference with cardiac muscle contraction, depolarization of the heart. So if you guys remember your little graphs from Biology 201, if you got a typical cell over here, the outside of the cell normally tends to be a little bit more positively charged than the inside of the cell, or the inside of the cell is a little bit more negatively charged. And that charge difference can be measured as a voltage that is below zero, because the inside of the cell is a little more negatively charged than the outside. And then you learned about depolarization, where you have these electrical changes along the membranes of neurons and skeletal muscle fibers, where temporarily that charge difference reverses, and temporarily the outside becomes more negative and the inside becomes more positive, and that's due to ion flow across the membrane. That's called depolarization when that happens. All right, so this happens in the heart in a rhythmic manner, which means it happens over and over and over and over and over again, following a certain rhythm. And it's also spontaneous. So that means it automatically happens. When you learned about skeletal muscle fibers depolarizing and having this electrical activity that leads to skeletal muscle fiber contraction, that was not spontaneous. Those skeletal muscle fibers have to receive signals from motor neurons to make that happen. That's not true in the heart. The heart will, on its own, generate its own action potentials, electrical changes, that then stimulate your cardiac muscle fibers to contract. Okay, now having said that, this automaticity or self-excitability that leads to action potentials being generated just spontaneously, that really only occurs in 1% of your cardiac cells. There are special cardiac muscle cells within the heart that do that automatically. So they automatically, on a rhythm, on a timed rhythm, they'll depolarize, repolarize, depolarize, repolarize. They do that automatically. They don't need any input from motor neurons. All right, and then we said we need that action potential to spread to all those cardiac muscle fibers that you have within the myocardium. That's where those gap junctions come in, those little pores. They allow those ions that are responsible for these changes in electrical activity to flow very quickly to the next cardiac muscle fiber, to the next one, to the next one, to the next one. And so you will very quickly have depolarization across the walls of the heart in unison. All these cells will do this at the same time. If you guys remember, there is an absolute refractory period, and with skeletal muscle fibers, that is where, all right, so when a skeletal muscle fiber depolarizes and then it repolarizes, there's a period of time where it can't do another action potential. That's called the absolute refractory period. The charge differences across the membrane have to be reset so that things can start working again. And same thing with cardiac muscle, but it's a longer period. It's 250 milliseconds, which is a quarter of a second in length. I know that doesn't sound like very long, but it's longer than what you have with your skeletal muscle fibers. All right, what this is showing you, this is a diagram from your textbook, and the blue line over here is showing you the electrical changes around the surfaces of cardiac muscle cells, like you see up here. These are the electrical changes. So these cells start out below -80 in terms of their voltage. So that's a little bit lower than what you have with your skeletal muscle fibers. Down more like -90 is where they start out, with -90 millivolts, and then you have this depolarization that takes place. So the voltage across the membrane goes up to +20 temporarily. Then you kind of have this slow repolarization that takes place at first, and then it goes back down more quickly, back down to -90. So those are the electrical changes that take place around the heart. There's a depolarization phase. Then this part is called the plateau, where it's kind of slowly repolarizing. And then phase 3 over here, you have a more rapid repolarization. And then your purple line over here, which I'll follow along in green. This is the tension within the cardiac muscle fiber itself. So just like what you have with your skeletal muscle fibers, those electrical changes around the surface of the cardiac muscle fibers leads to actin and myosin filaments, those proteins forming cross bridges and sliding across each other and generating tension within those cells. That's what they're graphing for you along the purple line. You know, this is more tension within your cardiac muscle fibers. This is less tension, and they're showing you how that times with the electrical activity around the heart. All right, so we're just going to kind of briefly hit some highlights there on that to show you how that's happening. We're not going to go back through cross bridging and you know, what happens as an action potential spreads across your cardiac muscle fibers. How does that in detail lead to actins and myosins cross bridging? Much of that story is very similar to what you guys learned with skeletal muscle fiber contraction. Calcium ions, though, are more involved with the depolarization process, whereas you learned more about sodium and potassium with skeletal muscle fibers. Calcium plays a much bigger role in that with your heart activity. But we are not going to go through all of those gory details. You can read about them in your textbook, though, if you would like some more information. Next video lecture, though, we will, I did mention we're going to discuss some differences and some of the key features of this electrical activity that are crucial for heart function, and we will pick up on those in lecture number 8.